Hello everyone, my name is Dr. Graham Cottrell and welcome to the second of the 3 videos in relation to molecules of life and in particular proteins. So the purpose of this short video is to introduce you to the 4 levels of protein structure and also discuss the types of chemical bonds that are involved in the formation of a protein structure, how a protein folds. So the first one is quite obviously called the primary structure, and this is just simply the sequence of amino acids. So we can see from the amino end the different types of amino acid. We have glycine, proline, threonine, glycine, and this is termed the primary structure, and it's just this sequence of amino acids. It's usually reported from the N-terminus to the C-terminus. And proteins, again, can be called peptides, polypeptides, or proteins. Very short stretches of amino acids, maybe up to say 50, are usually termed peptides. Anything longer, up to maybe 100, will be termed a polypeptide, and after that we refer to them as proteins. Although there's no set exact number for the differentiation between peptide, polypeptide and protein, but usually the smaller is a peptide and the larger one will be referred to as a protein. Secondary structure refers to the interaction of the amino acids, and it's usually to do with hydrogen bonds. So the most common forms of secondary structure are alpha helices, and here we can see these on the left. So the polypeptide chain wraps around itself and is linked together by these hydrogen bonds to form this helical structure. Beta sheets, again another form of secondary structure, and this is where you could see this could be the same polypeptide chain that's looped around, or it can be a different polypeptide chain where they go together in this sort of up and down, sort of almost like a folded sheet of paper. Again, these are held together by hydrogen bonds. So these are bonds within the same polypeptide chain, whereas beta sheets can be from different polypeptides, or it can be from the same one but just in a different direction. Tertiary structure is the overall shape of the entire protein molecule, and you can see here in the diagram the polypeptide chain loops round, it forms alpha helices here and here, and then we also have the beta sheets, but it's this overall fold of how the polypeptide chain interacts with itself, and the molecules will always bend and twist in such a way to achieve the lowest energy state. So the hydrophobic residues that we talked about in the previous video will be inside the molecule to keep them away from the water, so it'll be folded in such a way to minimize the interaction of hydrophobic molecules on the outside of the molecule. Other bonds that we will cover later that hold this protein shape together are the hydrogen bonds. You'll have van der Waals forces, hydrophobic interactions, ionic and disulfide bonds. So all these bonds interact to give a protein its shape. So what are hydrogen bonds? So these are formed, as you can see in the diagram here, between a hydrogen atom and a negatively charged oxygen on another amino acid. And it's this slightly positive. Remember that the oxygen will draw electrons towards it and become slightly negative, leading to a slightly positive or hydrogen ion. and it's this negative charge with the slight positive charge of the hydrogen that attract each other and hold the two residues together. We can also get hydrogen bonds not just with a negative charge, but also with a carbonyl group, as shown in the diagram with here with tyrosine. So it's that slight negative charge of the oxygen together with a positive charge on the hydrogen. And these can occur between not only the R groups of the amino acid, but also between the amino and the carbonyl groups. If the amino acid has a carboxyl group or an amine group, then it can also form what we call an ionic bond. So here we have the positively charged amine group attracting in the negative carboxylic acid group, and this group we refer to as an ionic bond. It's also formed based on an electrostatic attraction. Van der Waals forces. So this is a general term to describe an attraction between two molecules, an intermolecular attraction. These are generally much weaker than, say, hydrogen bonds or the ionic bonds, but there's generally lots and lots of them. So altogether, they contribute greatly to the folding and the holding of the protein structure together. And these are caused by a temporary dipole quality of the molecules involved. And this is obviously just due to electrons moving around. So that we have a slight positive charge at the end of one molecule is attracted to the negative charge on another molecule. Then we have hydrophobic interactions. As I mentioned a little bit before, that this is the driving force to keep the exposure to water a minimum. So here in the diagram we can see that we have the hydrophobic regions in red and the hydrophilic regions in blue. Then the molecules will pack in such a way to minimize the hydrophobic groups on the outside of the molecule, keeping them away from water. An example here is obviously deep inside a protein molecule where we have methyl groups which are hydrophobic, and they pack together and again have this weak van der Waals force. Then we have salt bridges, and salt bridges are a combination of two non-covalent bonds, and in this instance here we have a hydrogen bond and an ionic bond. So we have the hydrogen bond here of the alcohol group and a carboxylic acid together with an amino group and the carboxylic acid. So we have a hydrogen bond, an ionic bond, and together this is termed a salt bridge. As I mentioned previously, the amino acid cysteine can form disulfide bonds, and you can see here that within the single polypeptide, then these two cysteine groups can form a disulfide bond. So this occurs within the same polypeptide chain, and this will give an overall sort of fold to the protein structure. But it can also be used to link two polypeptide chains together. An example of these can be seen in the structure of an antibody or an immunoglobulin, which are held together by these disulfide bonds. So this is two cysteine molecules come together and form this sulfur-sulfur bridge. So the final level of protein structure is what we call quaternary structure. So many proteins that you will come across in this course are actually not single units. They are made up of different protein subunits. And it is the way that these different protein subunits interact with each other that we term quaternary structure. So the example here is hemoglobin. So this is made of different polypeptide chains, all folded, and they then come together to form the quaternary structure. And this quaternary structure again can be formed using different interactions, hydrogen bonding, disulfide bridges and salt bridges. So in summary, we have the 4 different levels of protein structure. We have the primary structure, which is the sequence of amino acids in the polypeptide. Then we have secondary structure, which is the formation of alpha helices and beta sheets. Tertiary structure is the overall fold of one particular polypeptide chain. And then the quaternary structure is different polypeptide chains coming together to form an overall structure. So I have a multiple choice question. This could be something similar to what is asked in an examination. So I'm just going to pause if you want to stop the video and have a go for this question yourself before I reveal the answer. So which of the following bond types are involved in the primary structure of proteins? Okay, so I'm now going to reveal the answer. Well, the answer, of course, is which of the following bond types are involved in the primary structure of proteins? So I'm now going to reveal the answer, and the answer is A, covalent bonds. So first we need to understand what is primary structure. But if you recall, the primary structure of a protein is its polypeptide, the sequence of amino acids in its polypeptide chain. And the amino acids themselves are held together by covalent bonds. So we're only concerned with the primary structure. So all these other types of bonds, the hydrogen bonds, ionic bonds, salt bridges and disulfide bonds, are not involved in the primary structure because that only relates to the amino acid sequence. And if you'd like to learn more about this, about protein structure, then in Molecular Biology of the Cell, Chapter 3, and in Biochemistry Stria, we have Chapter 2. Thank you all for listening.